Good afternoon. I'm Dr. Brian Shapiro, a Marketing Segment Manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It's entitled Advancements in Human Cell Line Cryopreservation for Assay-Ready Efficiency and is presented by Dr. Lucas Underwood and Ms. Diana Douglas. Dr. Underwood is a scientist with BioNexus Cryobiology at ATCC, while Ms. Douglas is a lead biologist in research and development at ATCC. In today's presentation, Dr. Underwood will highlight the challenges in generating working cell banks and post-thaw recovering cryopreserved vials. He will then go on to present a solution that allows you to go from frozen to data in one work day without the need for additional cell culture or expansion. Ms. Douglas will then show compelling data that demonstrates the utility of these assay-ready cells. She will show you that the thawed cells show high viability, fast recovery, and exhibit the ability to differentiate into macrophage-like cells that express the appropriate markers and functional attributes. If you have any questions for our speakers, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. Any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome our first speaker, Dr. Lucas Underwood.
Thank you, Brian, for the warm introduction, and welcome everybody to this webinar. I'm pleased and excited to present assay-ready cells today along with Diana. We'll start with a brief introduction to ATCC for those who are less familiar. ATCC was founded in 1925, and we're coming up incredibly close to an unbelievable milestone, ATCC's centennial birthday. ATCC is a nonprofit organization headquartered in Manassas, Virginia, with an R&D and services center in Gaithersburg, Maryland. ATCC is the world's premier biological material resource and standards organization featuring thousands of cell lines, tens of thousands of microorganisms, nucleic acids, and the media and reagents to support the research and usage of each. I also want to introduce a new initiative created by ATCC that is focused on developing new biological product formats that enhance user convenience. These formats are generally enabled by advanced biopreservation techniques. This initiative was realized by the creation of a team led by Dr. Nilay Chakraborty, whom has over 2 decades of experience in the field of cryobiology. Our team is now made up of 13 biologists, scientists, and engineers with a diverse set of scientific backgrounds and expertise. The primary focus of this team is the development and application of advanced biopreservation technologies and the accelerated embedding of those technologies into next-gen product development focused on user convenience and competence. This year, ATCC is launching 2 different series of products based on the advanced preservation technologies developed in this team. On the microbiology side, we've launched MicroQuant, a consumable quantitated lyopreserved pellet that is designed to simplify microbial QC testing. On the cell biology side, we have assay-ready cells, a consumable cell format designed to streamline and standardize cell-based bioassays, which of course is the subject of today's webinar. Here is our presentation overview. We'll start with some background and context focused on the importance of cell-based bioassays and specifically bioassays featuring immortalized cell lines. We'll also discuss cell culture as a fundamental technique required to perform these bioassays, along with a series of associated drawbacks. This will lead us into describing cell culture as an industry pain point and defining the need for a commercial solution to address these challenges. I'll then introduce ATCC's specific assay-ready solution and differentiating features. Then I will hand over to Diana to introduce our first 2 assay-ready conversions, present a catalog of application data utilizing these cells immediately post-thaw, and conclude with a summary and call to action for all of our listeners here today. So there may be no better crowning summary for the cumulative advancements made in the in vitro biospace than the FDA Modernization Act 2.0. This bill essentially refutes the Federal Food, Drug, and Cosmetics Act of 1938, which mandated animal testing for every new drug development protocol. Now, cell-based bioassays can replace the animal study requirement with the appropriate evidence and exemption status obtained. Cell-based bioassays can be used for a variety of different purposes in the biopharma and biotech space, and these are just a few examples, but are by no means a comprehensive list. They can be used for high-throughput screening of large chemical libraries for lead identification, mechanism of action studies, cytotoxicity and safety screens, and on the back end of development, cell-based bioassays are often employed for quality control and product release testing. There's a wide spectrum of biological models that can be used to facilitate these various use cases. However, as ATCC begins its journey into the bioconsumable space, we are starting with immortalized cell lines. Immortalized cell lines, while maybe lacking some of the more advanced biorelevance properties, have many critical advantages. These cell lines are often preferred for their ease of use. They don't require complex culture and handling procedures and are robustly viable with relatively simple equipment and protocols. These cells are globally available and most have been around for decades with an extensive array of published data for reference. Additionally, these cells are typically much more cost-effective than more advanced formats. And most importantly, if acquired from an authenticated source such as ATCC, the material is highly standardized and can be utilized to provide reproducible data when used for bioassays. The bottom line is that the insights gained from bioassays featuring immortalized cell lines build the foundation for subsequent testing in more expensive, less available advanced in vitro and animal models. Using these to narrow the scope of the required testing in more biorelevant models will result in a significant amount of time and cost savings. Now, let's take a step back and discuss cell culture. Cell culture is a fundamental technique that is generally a prerequisite to performing any bioassay. It provides a controlled environment that mimics natural in vivo conditions that facilitates the maintenance, growth, and eventual assessment of cells outside of the source organism. However, this ubiquitous technique comes with its drawbacks. It's time-consuming, where a majority of the time it's hands-off, waiting for growth or recovery of the cells. It's costly in labor, resources, and dedicated equipment, and promotes a certain level of resource dependence. For example, I'll cite the supply chain issues that most of us probably faced during the peak COVID years. Cell culture also promotes variability in the cell population over time, which can affect the accuracy and consistency of cell-based bioassays. This brings us to what I'll call problem one. Cell culture is used extensively for the traditional bioassay workflow, and its use can be separated into two phases. The first phase would typically be cell banking, which can take months of time to produce the material, cryopreserve it, and generally requires quality validation for the bioassay of interest. But this, of course, is preferred to the continuous culture alternative. The second phase consists of the actual assay workflow, which requires a short period of time of non-assay related cell culture to recover the cells thawed from cryostorage before directly inputting into the assay workflow. While a week or two may not seem like a substantial amount of time, this can quickly accumulate for repeats and long series of complex assay arrays. Note the section boxed in red, specifically the post-thaw recovery culture, stems from a secondary subproblem, which we'll call problem number 2, cryopreservation injury. Cryopreservation is an inherently unnatural and injurious process for cells, and the need for post-thaw recovery culture stems from that injury. There are several mechanisms that cause injury. Some have been studied more rigorously than others, but they can be organized into 4 general buckets: osmotic intolerance, intracellular ice formation or IIF, chilling injury, and cryoprotectant toxicity. The mechanism behind cellular osmotic intolerance begins when ice nucleates in the bulk cryosolution containing cells. It pushes the cells along with any other solutes into the intercrystal space. Effectively, you are removing the liquid solvent water from the solution in the form of ice, and the extracellular environment becomes hyperatonic, increasing the osmotic pressure to begin pulling water out of the cells. If If this is not properly controlled, extreme water kinetics can irreversibly damage cell membranes. And in terms of time, excessive overall water loss and shrinkage can cause the cells to undergo apoptosis. Now, some of this water removal from cells is necessary to prevent intracellular ice formation. For example, if the cooling rate for cryopreservation of cells is too fast, ample time is not given to remove enough water from the cells, and ice will begin nucleating inside the cells. These internal ice crystals can damage intracellular structures, puncture membranes, and disrupt normal biochemical processes. Third, what we often call chilling injury happens when cells are exposed to low non-freezing temperatures. Lipid biolayers become more rigid and they end up with metabolic imbalances with changes to temperature-dependent reactions. Certain cell types are more prone to this type of injury, such as monocytes. The last type of injury is caused by the attempt to address the other forms. We need cryoprotectants to reduce osmotic intolerance and IIF. However, certain cryoprotectants that can easily penetrate cell membranes can also disrupt several internal processes like cell metabolism and DNA packaging. If the concentrations and exposure times to these are not properly managed, it can also lead to the cell death that they were initially trying to prevent in the first place. Now, discussion of all of these potential injury mechanisms sets a gloomy picture. However, we all know that we can successfully cryopreserve with the traditional process highlighted here. We have our cells in culture, followed by harvest and replacement of the growth media with cryo media, and then aliquoting into vials and either freezing in a passive freezing container at -80°C or using a controlled rate freezer. And eventually we store for long-term in liquid nitrogen. Our traditional cryoprotectant is made up of some form of growth media, such as DMEM, supplemented with serum and 10% DMSO. Of course, it's nice to have a one-size-fits-all workflow and set of materials, but as we know, all cell types are different, and a unique set of process parameters and reagents gives us the optimal viability and function of cells. What we typically end up with using this standard workflow in cryomedia is a reasonably high percentage of viable cells post-thaw, but enough injury or disruption to the internal processes to require a period of cell culture for the cells to address those changes before optimal functionality returns. And that is essentially what we are trying to address. The solution to our defined problems related to cell culture is a consumable product that can reduce that requirement. However, one of the challenges in the market is not only are several terms to describe a similar type of product, for instance, Assay-Ready, Ready to Plate, Thaw and Go, just to name a few, but the actual format and deliverables of each of them is different as well. However, there are some key characteristics that these types of products have, where at least one is applicable. And these are saving time, reducing cost, and promoting consistency. So we have three different format definitions for Assay-Ready listed here. Starting from the left, the first is focused on assay preparation. These products are generally applicable and platform agnostic. The value proposition here is saving time and cost. Pre-plated frozen formats fall into this category, but these formats face challenges related to storage and stability due to the fact that they can't be stored at cryogenic temperatures. Other formats meant to directly replace the need for cell banking fall into this category as well. However, these solutions sometimes lack authentication and the proof of assay readiness. Some assay-ready products offer a complete solution for a specific assay. These typically feature engineered reporter cell lines and a set of companion reagents in a kit format. These products are optimized for consistency and response in a single assay and readout. However, due to the lack of versatility, it may be difficult to find a product solution that fits your workflow's specific needs. Last, we have a custom cell bank solution, creating seed or working cell stocks that can be directly used for bioassays. Typically, a customer will reach out directly with custom specifications. However, assay readiness is typically related to high cell density, and the cells may still require cell recovery and assay validation. This is not really an assay-ready consumable solution, but a request for custom cell banks from an authenticated bioproduction facility such as ATCC. Overall, each of these formats comes with limitations, such as the lack of versatility, long-term storage challenges, questionable quality and authentication, and the absence of a cryopreservation solution that minimizes post-thaw recovery. What we believe is that ATCC's assay-ready format features the best combination of beneficial features with minimal limitations. Now I'd like to introduce ATCC's assay-ready solution. Our slogan is frozen to data in 1 day because you can input these cells directly into your assay workflow immediately after thawing. This is a consumable vial format that can directly substitute for the need to generate cell banks for seamless integration into existing workflows. Unlike some of the other existing kit formats already discussed, these cells can directly input into a variety of assay workflows, and you'll see soon we provide a catalog of application data demonstrating several different use cases to increase customer confidence. You can expect the same biostandard quality that is typical of any product from ATCC, and we go even further with these consumable products, knowing the importance of reproducibility and consistency. Setting more stringent bioproduction controls compared to our standard products, and validating our manufacturing processes for precise inter and interlot specifications. From a technology and format perspective, what really sets this product apart is the unique cryobiology. Our team of experts developed an optimized cryopreservation medium and process that enhances the post-thaw recovery and allows our assay-ready cells to plug into assay workflows immediately after thawing. To conclude my section of the presentation, I'll discuss how we systematically optimize cryopreservation to reduce post-thaw injury. We target a reduced recovery time by optimizing the 2 major cryopreservation factors, cryomedia and freezing workflow. Now, the specifics are proprietary, but we can discuss the theory behind optimization. Cryoprotectants make up cryomedia can be divided into 2 categories: penetrating, meaning able to penetrate cellular membrane, and non-penetrating. Penetrating CPAs are going to be your DMSOs or glycerols that help prevent intracellular ice formation, while non-penetrating cryoprotectants like various sugars, proteins, and polymers, these are sometimes considered additives and help protect membranes and modulate ice crystal morphology and formation dynamics. The second factor is freezing profile. Every cell type in bulk solution has an optimal freezing rate to minimize injury and maximize recovery. We can fine-tune these rates with a controlled rate freezer and even introduce more advanced profiles besides a single linear temperature decrease. To summarize, we have an extensive pool of cryoprotectants with various known injury mechanisms. In some cases, we can narrow down formulation options with good background knowledge of specific cell reaction to freezing. And if we have certain target bioassays in mind, we can ensure they work effectively post-thaw. But when it comes down to it, eventually you have to perform the screens on a wide variety of potential components and formulations with different freezing profiles. Because as noted before, these 2 factors are not mutually exclusive. Our assay-ready cryomedia formulation is designed to be cell-specific. Just like optimal cell growth requires a specific media formulation, so does our cryomedia. Our media is animal byproduct free, which has certain regulatory benefits, but from a cryobiology perspective, it also increases consistency of outcome, particularly compared to formulations that contain serum. Because serum with known interlot variability can be magnified by freeze concentration that we discussed earlier with osmotic intolerance injury. A low toxicity formulation is particularly important for scaled up bioproduction where exposure times are going to be inherently longer. I'll conclude by emphasizing that this is a format technology. While we perform cell-specific optimization of cryopreservation workflows, the format description for the lab user is the same from product to product. A cryopreserved vial of cells that can be input immediately and directly into cell-based bioassay workflows immediately after thawing. And with that, I will hand over to Diana to introduce our first 2 assay-ready conversions with a heap of application data to demonstrate broad applicability for our cells immediately post-thaw. Thank you.
Thank you, Lucas. So Lucas covered ATCC's overall assay-ready solution. So now I'm going to share some of the process development and application data highlights for the cell lines that we currently have available. So the first 2 cell lines that we've converted into an assay-ready format are ATCC TIB-202, which is a THP1 line, and ATCC TIB-202-NFkB-LUC2, which is a THP1 luciferase reporter line. THP1 is a monocyte isolated from the peripheral blood of an acute monocytic leukemia patient. This cell line is extensively used to study monocyte and macrophage functions in vitro as an alternative to human PBMC-derived cells. The luciferase reporter cell line, which was derived from the parental cells, expresses a firefly luciferase gene under the control of an NFkB promoter. And this line is useful for monitoring the activity of interferon gamma-induced signaling pathways. A few points I wanted to highlight before we get into the cell line specific data. So our assay-ready cells have been carefully created to exhibit high viability, as well as low variation and low variability, both inter and intra-lot. And they have growth and morphology characteristics very similar to that of the parental cells. So these cells are ready to use directly from thaw, so you can complete your assays and collect data in one workday. So some examples of the application data that I'm going to go over today include differentiation to macrophage-like cells, morphology of these differentiated cells, qPCR, flow cytometry, phagocytosis assays, and luciferase assays. A key aspect of assay-ready cells is their high post-thaw viability, and on top of that, a reliable and repeatable high post-thaw viability. So the data on this slide summarizes the high viability of our cells, as well as the consistency of this data. So like I mentioned, our cells are ready to use immediately after thaw. And to confirm this, we tested about 75 vials in product development. Our internal design goal is 80%. And if you look at this slide, so we have the THP1 assay-ready cells on the left and the LUC2 line on the right. So all of the vials tested in 3 batches by 3 different operators have an average viability that easily surpasses 80%. The majority surpass even 90%. So we're quite confident in these results and their repeatability and in their ability to aid scientists in obtaining consistent and high-quality assay results. So not only do our assay-ready cells show consistent high viability immediately post-thaw, these cells retain those attributes after they have been stored in vapor phase liquid nitrogen. So thus far, we have stability data up to 6 months. However, these are a new product, so this is something we are continuing to monitor. And we will update this information as new data is available, as we do expect these cells to retain a high viability and functionality significantly beyond 6 months. And this is something that you don't necessarily see with the pre-plated assay-ready cells. They often have a much shorter and very specific shelf life. So again, this is just underlining the versatility and the flexibility of our vialed assay-ready cells. Another key aspect of our assay-ready cells is their similarity to the parental or non-assay-ready counterparts. So here we're looking at images of 3 batches of assay-ready cells in comparison to our control. And we see no differences in the morphology or the growth characteristics of the assay-ready cells. All the cells are suspension monocytes as expected. So in addition to the usual data and instructions provided with all ATCC cells, we're including 2 companion protocols with the assay-ready cells. So the conditions in these 2 protocols have been optimized across both R&D and manufacturing to ensure high-quality cells and high-quality results. So first, we have a step-by-step thawing protocol specific to ATCC's assay-ready cells. So as seen in this slide, this protocol provides instructions for the best way to thaw cells to ensure that they are healthy and ready to go. So we also developed a differentiation protocol. So as we all know, monocyte to macrophage differentiation is one of the most commonly used and physiologically relevant applications for THP1 cells. And using PMA is a very well-established method for differentiation. But if you actually look at the literature, the precise practice and method varies hugely from lab to lab. So therefore, there's also a great deal of variation in the end product of cells. So the phenotype and genotype of the macrophage-like cells that labs end up with also varies greatly. So this is a protocol that we've optimized for our assay-ready cells so that customers can be confident that they are obtaining macrophage-like cells that offer reproducible functionality. So our protocol details a 72-hour PMA-induced differentiation process, and this will be included as an insert with all of our cells. The images on this slide show some examples of what you can expect to see as cells go through the differentiation process. The images in row A are our parental or non-ARC cells, and the images in row B are the assay-ready cells. Again, just to highlight that you can expect the same morphology and same behavior in both lines. Within the first 24 hours of treatment, cells will adhere to the plate, and throughout the next 48 hours, cells will continue to undergo morphology changes as they differentiate. You'll see that the cells become enlarged, specifically their cytoplasmic volume increases, and the granularity of the cells very obviously increases as well. These are hallmark characteristics of macrophage-like cells, and some examples can be seen in the HiMag image on this slide. Additionally, beyond our differentiation platform, cells can be further differentiated into subsets of classical macrophages or dendritic-like cells, depending on your lab's specific interests. We also use these differentiated cells in a number of application assays to ensure that the cells are expressing the appropriate markers and functionality. So first, we're looking at mRNA expression of CD14 and CD36 by qPCR. So CD14, which is expressed on the surface of monocytes and macrophages, is often used as a key indicator of THP1 cell differentiation, and its expression can increase with the induction of PMA. CD36 is a scavenger receptor. It contributes to the uptake of oxidized LDL particles and fatty acids by macrophages, and it participates in the internalization of apoptotic cells. CD36 is also known to be regulated during THP1 monocyte differentiation. So to evaluate these key marker gene expression differences between parental THP1 cells and our assay-ready THP1 cells, as well as their differentiated macrophages, qPCR was carried out. So what we found was that both the parental THP1s and the assay-ready cells show low-level expression of CD14 and CD36, but their expression is dramatically increased in the differentiated macrophage-like cells upon PMA stimulation, demonstrating that the THP1 assay-ready cells maintain the key marker expression characteristics of the parental THP1 cells. We also designed a flow assay to look at the surface expression of key markers. So like I mentioned, CD14 is one of the hallmark markers that researchers look at when they're evaluating THP1 cells and differentiation. But there's so much variability when you look at different cells from different sources or even different data sets from the same cells. There are actually two forms of CD14 which may account for some of these differences. So although it was initially characterized as a cell membrane protein, CD14 exists in both soluble and membrane-associated protein forms. So flow cytometry analysis was carried out to evaluate the cell surface CD14 expression in parental cells and assay-ready cells, as well as their differentiated macrophage-like cells, as seen on this slide. So CD14 was expressed on almost the entire cell population of the parental THP1 cells and assay-ready cells, as well as their differentiated macrophage-like cells. So with our assay-ready cells, you're getting cells that have been thoroughly authenticated and characterized. In addition to marker expression, our assay-ready cells also retain the functionality of macrophages. So phagocytosis of pathogens, apoptotic cells, and debris is a defining feature of macrophage function. The phagocytic capability of THP1 assay-ready derived macrophage-like cells was assessed using photobioparticles from Thermo. So these particles only fluoresce when they're internalized in the acidic environment of the phagolysosome. We also used lysotracker from Thermo to stain lysosomes, which accumulate in the cytoplasm with macrophage differentiation. So what we're looking at on this slide is the ingested red photobioparticles in cells undergoing phagocytosis in B. Then in C, we can see that the cellular lysosomes are stained green by lysotracker. Then in figure D, we see the overlay and the colocalization of the ingested red photobioparticles in the cells undergoing phagocytosis with the cellular lysosomes that were stained green by lysotracker, indicating phagolysosome formation during the phagocytosis. And these data confirm that THP1 assay-ready cells can effectively differentiate into macrophage-like cells and maintain phagocytic capacity. So to change gears a little bit and look at the luciferase line. So for the TIB-202-NFkB-LUC2-AR cells, we focused obviously on luciferase assays for our application data. Again, these cells were assayed directly from frozen. Very briefly, they were treated with 50 nanograms per mil of LPS to activate the NF-kappa B pathway. Bright glow was then added and luminescence was read after about 15 minutes. So on the left, we saw that all 3 batches showed a very robust response with at least a 200-fold increase in luminescence above background level when stimulated within 8 hours post-thaw. And then the graph on the right shows that this response can be expected at any time within an 8-hour workday, just again providing some flexibility with your assays. In addition, we also generated dose-dependent response curves for the assay-ready cells. So this slide shows these response curves looking at bioluminescent signal intensity after treatment with a series of concentrations of either LPS or LPS and Bay11. So LPS, of course, activates the NF-kappa B pathway, while Bay11 is an NF-kappa B inhibitor. So for LPS treatment, we see the same positive response curve in both the parental and assay-ready cells. And then we see the inverse for the inhibitor in part B. And this type of drug response experiment could be utilized, for other drugs or compounds of your choice, depending on your lab specific interests. Now, I've just covered quite a bit of information and application data in the past several slides, so I just want to recap and provide a quick data summary. So cell expansion has been optimized by ATCC and cells have been frozen with a proprietary animal byproduct-free cryomedia. These assay-ready cells provide high post-thaw viability and cell count, as well as authenticated cells for reliability and repeatability. The THP1-AR and THP1-NFkB-LUC2-AR cells have been extensively tested in application assays and show the appropriate marker expression and functionality. So the TIB-202-AR cells can be differentiated into macrophage-like cells. We also have a protocol that will be provided with the cells. These cells exhibit hallmark morphological characteristics of macrophages. They express increased levels of CD14 and CD36 upon PMA activation, and they retain phagocytic capabilities. The TIB-202-NFkB-LUC2-AR cells, they have a robust luciferase expression upon LPS stimulation. They also show appropriate drug response curves. So overall, ATCC's assay-ready cells are a direct substitute for cell banks and can be input into bioassay workflows immediately post-thaw. If you would like more information on our assay-ready cells or any of our products, please visit our website at atcc.org. And we would love to hear some feedback after this presentation regarding what cells your lab would like to see considered for addition to the portfolio. So thank you so much for your time, and we'd be happy to answer any questions.
Well, thank you, Diana and Lucas, for the excellent presentation. In just a few moments, we will begin our Q&A session. Please remember to use the chat function available in the webinar program to submit your questions. And it looks like we've had a whole bunch of questions come in already, actually. So let's see. I'm going to cherry pick and kind of start with some of the more general questions around the product format, and then move over into some of the scientific questions. So first of all, the first question's around packaging options. Could either Diana or Lucas, you talk about the packaging options for the assay-ready cells?
Sure. There are several available packaging options. You can buy them as a single vial. There's also a 3-vial pack, a 5-vial pack, and a 10-vial pack.
All right, and that's for both versions of the assay-ready cells, the luciferase reporter and the, okay.
That's correct.
Excellent. Excellent. And someone is wondering how the assay-ready cells are produced.
Yeah, I can take this one.
Okay, yeah.
Sorry. So yeah, these cells are cultured and banked at scale, and following a significant quality testing to ensure the stability and functionality for extended periods. As mentioned previously, during development, optimizations were performed for production processes and cryopreservation to improve the inter and interlock consistency of the vials.
All right. Speaking of consistency, and actually this is maybe more of a storage question, we do not have enough liquid nitrogen vapor phase space. Can we store the assay-ready cells in LN liquid phase? And then, a flip side to this, can you store the cells in -80 degree storage?
We did take a look at this, and cells can be stored in the liquid phase of liquid nitrogen if you don't have enough vapor phase. We would not recommend -80 degree storage, however.
Right, and there's a significant loss in the product viability or functionality, rather, if it's stored in the -80 degrees, correct?
Yes, that is correct.
Right. So I guess that really leads into the next question of any specific handling instructions.
So there are, in addition to the normal product information sheets that come with every ATC cell line, there are specific thawing and handling protocols available with all the assay-ready cells. So these protocols are available on the assay-ready cells product page on the website.
Okay. I guess one critical thing to share, though, would be around what to do when the cells come into your laboratory, right?
Yeah, they should immediately be stored in liquid nitrogen.
Right, as soon as possible.
Yes.
Okay, and you guys have shown a lot of data around this in this webinar, but how do assay-ready cells compare to cells prepared from culture?
Well, the cryopreservation reagent allows the assay-ready cells to be used at thaw with no loss of function and no need for recovering or additional culture. So we did test the assay-ready cells in multiple assays along with freshly cultured cells, and they did perform similarly. And there is a lot of reference data in the form of technical data sheets and application notes, et cetera, again, found on the assay-ready cell product page on the website.
Nice, nice. All right, a few more questions kind of around use of the product have come in. Is there any difference between the standard parental versions of the cell lines and the assay-ready version with regards to, say, genome editing or engineering?
Yeah, that's a good question. No, so there's no transient or long-term genetic engineering performed to the cells to improve the post-thaw performance. So like the only changes that are made are to the manufacturing process and additionally the cryomedia for usage immediately post-thaw.
Okay. Now, could I use the standard version of the cells, the parental cell lines, immediately post-thaw, like the Assay-Ready format?
Yeah, so we do not recommend it. There's anecdotal evidence to suggest that, you do need some period of time for post-thaw functionality to return. So additionally, there is greater lot-to-lot and intra-lot variability for the standard version of the cells compared to the assay-ready version. And, as Diana has mentioned a couple times, these have been extensively validated for immediately using them post-thaw. So hopefully that can help reduce some of the internal validation requirements in your respective labs.
Okay. Now, say you have some cells that are left over from your experiment. Can you keep them in culture?
So the short answer would be yes. There isn't any kind of kill switch that would make them completely unusable, but we highly recommend not doing so. These are meant to be a consumable product, and they've been validated as such for assays very soon after thawing. The continued culture could result in inaccurate or inconsistent bioassay results. Again, so we would not recommend continuing to culture them and use them.
Gotcha. Wow, we've had a lot of questions come in. This is good. This is great. So what are the release tests performed on these assay-ready cells?
So the lot release criteria for every lot of cells is viability, cell number, and for the luciferase line, luciferase assay. And that's in addition to all the standard internal QC tests that ATCC always performs, including an STR profile, VAC-T, virus testing, mycoplasma testing, and CO1 testing.
Okay, great. So for the animal-free format of the cells, is an alternative serum used to support the cells while frozen? That could be
I'll give it a shot. So this is a completely animal byproduct-free formulation. Although it is proprietary, so we can't speak to it too much, but there are alternative cryoprotectants included that are serving the purpose of serum. That helps.
Okay, that'll work. Does the application data generated from post-freezer or 6 months stored ARC, hold on. I guess, how long have the assay-ready cells been frozen? Do we have any data that's like, from the assay-ready cells that have been frozen for like 6 months, say?
So most of the application data is from cells frozen more short term. We do have some stability testing at 6 months just to ensure that the cells are retaining, their viability and their cell count, et cetera. But the majority of like the assays that were performed in the application data are on more short-term frozen cells.
Okay, great. is there a plan to make other other cell types in an assay-ready format? And I think this is a good one for Lucas to speak to.
Yes, so as I mentioned, this is a format technology, so the plan is to expand into further extensions, further cell lines. However, we don't have the specifics for those currently available.
All right, good, good. Now, how many cells come in one vial?
They're guaranteed to have a minimum of 6 million cells per vial.
Would you want to use these cells for a transfection experiment?
I mean, you could use them for really any, type of assay you think would be I guess basically the short answer would be sure. I think they can be used, they're suitable for, various types of assays. In the application data, we did test a number of assays, but you would want to, first verify the compatibility with your specific experimental requirements. Before
I just don't see these cells for like, long term, but maybe for a transient transfection or
right.
or shRNA or ASRO or something like that. But I wouldn't see using it in a extended, experiment.
Right.
All right. And then a customer is interested in the cost of the products, which we will reach out to you directly to share that information. However, it is on the web on ATCC's website at www.atcc.org/assay-ready. Let's see. Okay. Have we compared our assay-ready cells to a competitor's assay-ready cells head-to-head? And do we have any competitors that actually have an assay-ready cell exactly like ours? For example, many competitors, their idea of assay-ready is just a vial that has like 6 million cells in it or something like that, or they may have some reporter in it, so it's specific for an assay. Is there anyone that has something that's like cryologic in nature like ours is, rather than just a whole bunch of cells, or as part of a kit with a bunch of plates and reagents?
Lucas, do you want to take that one?
And no, and I don't know is a perfectly good answer, by the way.
Yeah, I would agree. I think, Brian, you were kind of alluding to it. There are a lot of different sort of versions of assay-ready, and so finding a real direct comparison is challenging, particularly for a specific cell line. So at the moment, we don't have any direct competitor analysis to present, but you may look into that.
All right, good, good. Let's see. I am running through the questions, and I feel like we've got a lot of duplicates that have come in. People are more or less asking the same question. Let's see. Now, the protocol for TIB-202 cells, does that work for the NFkB cells? I guess that would be the thawing protocol.
Yeah, the thawing protocol is the same for both.
Gotcha. How long can the assay-ready cells be maintained once thawed? And be maintained, I would say, probably in just one experiment. Like, what would the longest experiment be?
While we were doing application data, like I probably had cells, in culture for about a week. I don't know that I went much past that. It just wasn't really in, the timeline of the experiments that I was doing. So I can't say like there's a, exact like end date when the cells will stop, working. I mean, they're meant for short-term, fairly short-term use, one experiment basically. And like I said, for the application data, most of our experiments were, about a week long.
Gotcha. For that PMA differentiation protocol, that works for the NFkB cell line as well, right? With regards to the number of cells per well?
Yeah, you could use it for those two cell lines as well, yeah.
Okay, great. All right, here's a chance to sort of repeat a critical point from earlier. What's the maximum time to store cells in -80 degrees without affecting them?
Oh, I wouldn't store them at -80 at all. I would say as soon as you get them, immediately store them in liquid nitrogen just to ensure there's no loss of, viability or functionality.
Right. Yeah, it is super critical to, as soon as these cells get into your laboratory, put them on liquid nitrogen vapor phase.
Mm-hmm.
Okay. Well, it seems like we've hit on all the questions. The rest of them have come in, are pretty much duplicates. So I would like to thank both of our speakers, Lucas Underwood and Diana Douglas, for the excellent presentation, and thank you everyone for attending. If you want to learn more about our assay-ready cells, you can go to www.atcc.org/assay-ready. You can come and talk to us in person about assay-ready cells as well if you're attending the Society for Immunotherapy of Cancer annual meeting in Houston, Texas, or at the Society for Laboratory Automation and Screening in San Diego, California. Hope to see you all there. Thank you, everyone, and have a great day.